Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Srivastava. Today my presentation title is Seed Germination. Before presentation I would like to introduce myself. I am Vijay Kumar Srivastava. I have done MS Agriculture with a specialization in agronomy from GB Pant University of Agriculture and Technology, Pantnagar in 1996. So let's move to the presentation Seed Germination. What is seed germination? Germination is the ability of a seed when planted under normal sowing conditions to give rise to a normal seedling and developing into new plant. The process of seed germination starts with the imbibition of water by seed coat and emergence of a growing root tip of embryo. The process ends with the development of embryo into a seedling. So here we have shown the different pictures which represents the germination process. Here to study the types of seed germination. The germination is of two types. First one is epigeal germination and second is hypogeal germination. What is epigeal germination? In this type of germination, the hypocotyle elongates rapidly and arches towards pulling the cotyledon which moves above the soil. Here we have shown the germination of cotton seeds where the seed coat is coming out along with cotyledon. And second type of germination, hypogeal germination. In this type of germination, the epicotyle elongates and the cotyledon remains below the soil. Here we have shown the germination of maize seed where the cotyledon is inside the soil and the radicals are moving inside and plumule is coming outside without cotyledon. Now to discuss germinating seedlings. After germination, there are four types of seedlings which are observed. Like first one is normal seedling, second is abnormal seedling, third is dormant seedling, and fourth is dead seedling. Now what is normal seedling? These are the seedling which will show the capacity for continued growth and development into normal plant under favorable conditions of water supply, temperature and light. These are the seedlings which are possessing all the essential structure to meet the seed requirement. And abnormal seedling, these are the seedlings which do not show the capacity for continued development into normal plants even provided favorable conditions. And this type of seedlings includes damaged seedlings, delayed seedlings, deformed seedlings, etc. Third is dormant seedling. These are the seedlings which are germinating and stops further developments, and these are no capacity to grow into normal plants. And fourth is dead seedlings. These are hard seeds which just going for the water soaking and there is no sprouting and no germination at all and seed remains the same. So these are the dead seedlings. Now to study the ideal conditions which are needed for germination. Germination is depending on various factors, like first one is optimum moisture. This is one of the primary requirement for germination. If optimum moisture or soil water is available in the soil, there will be imbibitions of water by seed coat and rupturing will take place and there will be better germination conditions. Second is optimum temperature. This is also one of the important factor and there is correlation between moisture and temperature. If temperature is optimum along with moisture, then there will be good germination. Then sunlight. Sunlight is also affecting the germination in a different way, like it is affected by red lights and blue lights. If red light is there, it will promote the germination and blue light inhibits germination. After that, aeration. Aeration where the presence of oxygen is also needed for the biological activity of the seed, biochemical activity, and it will improve the germination. After that, nutrient rich soil. If soil is good and it is providing the all favorable conditions to the seed, there will be definitely good germination. After that, good quality seed. This is the inherent character of the seed. If it is having the good genetics, good quality, then it will improve the germination status. If there is some issues with seed, then we can go for some chemical treatment which will improve the germination Like generally we are following the ethanol treatment which is used for seed germination improvement, and the chemical which is used ethanol which is 2-chlorostyl phosphoric acid. And by the soaking of the seeds with 25 to 30 ppm solution in ethanol solution with 40% volume for 6 hours will improve the germination. Now we will study about factors affecting germination. Germination is affected by soil as well as environmental factors about which we will study here. First one is soil. In this group, the soil type, soil texture, soil structure and soil microorganisms greatly influence the seed germination. If soil is having good quality, it would have good water holding capacity, optimum soil temperature, aeration, which will favor the seed for germination. Now second is environment. Generally the environmental conditions favoring growth of seedlings and also favors germination. In case of standing crop while cultivation, germination doesn't occur in harvested seeds until the seeds attain physiological maturity. So while harvesting, a special care should be taken that harvest should be only after attaining the physiological maturity to use the grains or seed to take the next crop. After that, water, which includes soil moisture and seed moisture. Imbibition of water is the prerequisite process for germination. Means after absorbing the water, the seed coat will swell and it will rupture and it will allow the germination. Slower rate of germination is noticed in places where soil moisture is near or at wilting point. So insufficient quantity of moisture, there will be fast germination, while there is a stress of moisture, there will be delay or less germination. After that, temperature. The optimum temperature is that one gives the highest germination percent in the shortest period of time, and it is having the correlation with soil moisture. Next is light. The most effective wavelength for promoting the seed germination is red lights having the wavelength of 660 nm and the blue lights having the inhibiting germination character having the wavelength of 730 nm respectively. Now atmospheric gases. Most crop seeds germinate well in the ambient composition of air having the oxygen of 20% and carbon dioxide of 0.03% and nitrogen of 78.2%. After that, exogenous chemicals. There are some chemicals which induces or favor quick or rapid germination. So these are the factors which affect the germination. In case of exogenous chemicals like ethanol solutions we are using, after which the seed soaked in ethanol solution, there will be pickup and fast germination. Our next studied point is germination testing. Germination is defined as the emergence and development from the seed embryo of those essential structures for the kind of seed sample submitted, which indicates its ability to produce a normal plant under favorable conditions. The objective of germination test is to determine the germination potential of a seed lot. The percent germination is the estimated viability of a seed lot tested under specified conditions and within a specified period of time. Each test must consist of 400 seeds which are drawn from the working sample and then randomly divided into 4 replicates of 100 seeds or 8 replicates of 50 seeds. Now turn to study the emergence of seedling out of the seed coat. Germination results in splitting of seed coat and emergence of the growing seedling. The radicle comes out first and grows downward, and then plumule comes out and grows upward. Due to the continued growth of this seedling, the plumule comes out of the soil, exposed to light, and develops its own photosynthetic apparatus. A splitting of seed coat may take place either by imbibition pressure or by internal pressure created by the growing primary root or by hydrolytic enzymes which act on cell wall contents of seed coat and digest it like cellulose and pectinase enzymes. Sometimes the seed coat may extensively rotate by the activity of microorganisms in the soil. Here we have shown the pictures of common garden bean and maize crop and how the germination process took place. The radicle comes first, where the roots are going downward and the plumules are coming outside. Here to study the dormancy of seeds. What is seed dormancy? Seed dormancy is defined as a state in which seeds are prevented from germinating even under environmental conditions. It is for some duration. As per the genetics, the seeds are containing the dormancy period. After passing that period, the seeds will germinate. Then there will be no issue. All the viable seeds have capacity to germinate if placed under suitable conditions necessary for germination. But some seeds fail to germinate sometimes even if placed under the condition favorable for germination. This may be due to some internal factors or due to a specific requirement for some environmental factors. During this period, the growth of seed remains suspended and they are said to be rest stage or dormant stage, and this phenomenon is called as dormancy of seeds. Here we have shown the picture which represents the normal seedlings where the germination is excellent, very good, and in some cases if dormant seed is there, this germination will be very poor because it takes time. As per genetic characters, the germination will improve after resting phase is over. What are the factors which are causing dormancy of seeds? There are various factors, like first one is seed coats impermeable to water. The seeds of certain plants, especially those belonging to the family Leguminosae, Solanaceae, Malvaceae, etc., have very hard seed coats which are impermeable to water. The seeds remain dormant until the impermeable layer decay by the action of soil microorganisms. So, because of hard seed coat, there will be less imbibitions or absorption of water, which will delay the germination and it will cause the dormancy of seeds. Second is seed coats impermeable to oxygen. In many plants such as cockleber and many grasses, the seed dormancy is due to the impermeability of the seed coat to oxygen. However, during the period of dormancy, the seed coat gradually becomes more permeable to oxygen so that they may germinate. Third is immaturity of embryo. In certain orchids and some crops, the seed dormancy is due to the immaturity of the embryo, which fails to develop fully by the time the seeds are shed. In such cases, the seed germinates only after a period or a rest during the which the development of embryo inside the seed is completed. Fourth factor is germination inhibitors presence. In certain seeds, the dormancy of seeds is due to the presence of certain germination inhibitors like coumarin, ferulic acid, abscisic acid, etc. These may be present in endosperm, embryo, testa or juice or pulp of the fruits, which inhibits the germination process. After that, fifth is absence of growth promoters. In certain seeds, the dormancy of the seed is due to the absence of certain growth promoters. Then sixth is chilling or low temperature requirement. In certain plants such as apple, rose, peach, etc., the seeds remain dormant after harvest in the autumn as they have a low temperature or chilling requirements for germination. In nature, this requirement is fulfilled by the winter temperatures. In such case, the seeds remain dormant throughout the winter season and germinates only in the following springs, which is the genetic character of the seed. Then seventh factor is light sensitive seeds. In many species, the germination of the seeds is affected by light resulting in the seed dormancy. Such light sensitive seeds are called photoblastic. Seeds of lettuce, tomato and tobacco are positively photoblastic and germinates only after they have been exposed to light. On the other hand, the seeds of certain plants are negatively photoblastic, means they are having the negative reaction to light and their germination will be inhibited. Like red light is promoting the germination and blue light inhibits germination. Now eighth factor is nature of seed for periodical dormancy. This is the genetic character of seeds having the dormancy which breaks automatically after completing the rest phase or dormancy period. Now here to study the dormancy treatment methods which are resulting in breakage of dormancy. Like first one is escarification, where the seed is treated by escarification with sand, knife and sandpapers. Then temperature treatment, where the high and low temperature is given to the seeds for a certain period, certain time, which may result in breakage of dormancy. After that, light treatment. The red light is known to improves the germination, so red light treatment will be helpful in breaking of dormancy. Then pressure treatment, chemical treatment, where the seed is soaked with potassium nitrate solution, hydrogen chloride, hydrochloric acids and sulfuric acids or gibberellic acids and ethanol solutions. After that, cool treatment is there, where the chilling temperatures are given to some crops, some seeds, which are resulting in breakdown of dormancy. During germination process, there are some physiological and biochemical changes which are taking place in seed, about which we will study here. Like first one is water uptake. Seed germination starts with the imbibitions, means absorption of water by dry seed coat. Due to imbibitions of water, the seed coats become more permeable to oxygen and water because of swelling and absorption of water. Then it becomes less resistant to outward growth of embryo. So the soil moisture or soil water is playing important role for the germination of the seed. After that, respiration. There will be rapid increase in respiration rate of embryo after providing the water to the soil and availability of soil moisture, which correlate with temperature. Sucrose is probably the respiratory substrate at this stage, which is provided by endosperm. And third is mobilization of reserve materials. As germination progresses, there is mobilization of reserve material to provide building blocks for the development of embryo, energy for the biosynthetic process, and nucleic acids for control of protein synthesis and embryonic development for the growth of plants. So these are the chemical and physiological changes taking place while germination. After germination, there are some changes are taking place in components or compounds of seed. Like first one is changes in nucleic acid. In monocots, during imbibitions, there is a rapid decrease of DNA and RNA content in the endosperm with a simultaneous increase in the embryonic axis, probably due to their transportation as such. High concentration of RNA ribonucleic acid in the embryonic axis precedes cell division. Due to more cell division, diribonucleic acid DNA content is increased. Then second is carbohydrates. Insoluble carbohydrates like starch are the important reserve food of cereals in the endosperm. During germination, a starch is hydrolyzed first into maltose in the presence of alpha amylase and beta amylase, and then maltose is converted into glucose by maltase enzyme. The glucose is further converted into soluble sucrose and transported to growing embryonic axis. During germination, the embryonic axis secretes gibberellic acids into the aileron layer, which causes synthesis of alpha amylase. Now changes in lipids. Plants like castor bean, peanuts, etc. store large amount of neutral lipids or fats as reserve food in their seeds. During germination, the fats are hydrolyzed into fatty acids and glycerol by lipase enzyme. Fatty acids are further converted into acetyl CoA by the process beta oxidation. The acetyl CoA is further converted into sucrose by gloxylate cycle and is transported to the growing embryonic axis. After that, changes in protein. Some plants store protein as reserve food in their seeds in the form of aileron grains. Protein are hydrolyzed into amino acids by peptidase enzyme. The amino acids may either provide energy by oxidation after deamination, which is known as removal of amino group, or may be utilized in the synthesis of new proteins. Fifth is inorganic material changes. A number of inorganic materials such as phosphate, calcium, magnesium and potassium are also stored in seeds in the form of phytin. These stored materials are liberated during germination due to the activity of various phosphates including phytase. So these are the changes which are taking place in seed components and compounds during germination. So this presentation was all about seed germination. Hope this presentation will be very helpful to all of you. Now my presentation ends here. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Srivastava. Having request, please visit the channel, subscribe it and provide your kind and valuable feedback for further improvements in next coming presentations. Thank you.